of important ways. One is it doesn't seem to be addictive at all, according to a study out of Harvard. But it has some other very important effects, some of which mimic nicotine. Here's one of them, which doesn't mimic nicotine, but it's perhaps more pronounced with anatobine. When you see this number up here, RCP006, that's just our internal designation for anatobine. But what we've done here is we've inflamed some cells, and then we've added progressive amounts of anatobine. And what you can see is this reduction in inflammatory cytokines. We're actually measuring something called NF-kappa B. NF-kappa B is a sort of a master controlling protein that controls inflammatory processes in many cells. And so it's very important to see this anti-inflammatory effect of anatobine. And it does something else very interesting too. It cuts down the amount of base messenger RNA. Let me explain what that is. What we've done here is we've increased the amount of inflammation in some human neurons. And what you see here is an increase in the amount of messenger RNA. That's the message that comes from the nucleus to say to the cell, make some more protein. And the protein that it's saying should be made is base, one of those pairs of scissors. So if you make more of that enzyme, that cutting enzyme, you'll make more amyloid. If you make more amyloid, you make more Alzheimer's. And what you can see is when you add anatobine, you really suppress that effect very dramatically. So anatobine has the ability to suppress the pro-inflammatory, but also the pro-Alzheimer effect in human neurons. And here's some confirmation of that. This just shows you that the amyloid levels are going down as we increase the amount of anatobine. There's basically two types of amyloid. This one is a little bit more friendly than this one. This one's called 1:42, and it's the really noxious, really toxic form. This one is less toxic. And some people really think that it's the ratio of 42:40 that determines how much damage gets done in the brain. But either way, with anatobine, you can see this reduction in amyloid production. So this was all preclinical work and very comforting and very interesting. But it was in cell culture. We really wanted to know, could we see the same thing in the brain? And here we have a mouse brain, in fact, that has inflammation, a lot of inflammation. And here's a mouse that's been treated with anatobine. And again, you can see this reduction. So that the idea is that this anatobine can very easily get into the brain and suppress inflammation and suppress the production of amyloid. So of course, we and others are thinking that this might be something that we can, that might be beneficial in Alzheimer's. I will just explain one thing though at this point, and that is this is being developed as a nutraceutical, not as a drug. And there's a big difference. There's a huge difference as far as the FDA is concerned. There's a big difference in terms of regulation, and there's a big difference in terms of cost. To give you an example, just filing what is called an investigational new drug application for nilvarapine cost us at least $10 million, and that was just to produce the paperwork and some studies. One doesn't need an IND with a nutraceutical, so you can get into clinical trials or clinical use or just general use much more rapidly. However, the FDA has very, very strict rules about what is a nutraceutical and what isn't. And if you try and modify diseases, then the FDA regards that compound as a drug, essentially. So that we can't advance this as we normally would as a drug, but nevertheless, these are very interesting preclinical findings, and this is a compound that's actually available already on the internet and elsewhere. But this, I think, you might find interesting. Here are those two. Here are mice, one treated with anatobine, one treated with placebo. And I can tell you, because you probably can't see it, that the hidden platform is about there. It's about there in both cases. And these are transgenic mice. So these are mice that by the time they were asked to, by the time we put them in this test, maybe we did ask them, we're very kind to our mice at the Institute. By the time they agreed to do this test, they had a lot of Alzheimer pathology. They were quite old, in fact, and had a lot of amyloid pathology. And so you can see